We're being recorded.
Yeah. All right. Well, I'm happy to introduce our speaker today. Melanie During is a paleontologist at the start of a very promising career. She's made a remarkable discovery that the meteor that ended the age of dinosaurs impacted the Earth during spring in the Northern Hemisphere 66 million years ago. And so we're pleased to have her here to tell us at firsthand about this study on which she was the lead author. This work was part of a master's thesis in Amsterdam, for which she won the 2018 Escher Prize of the Royal Dutch Geology and Mining Society. She's now a PhD student at the University of Uppsala in Sweden. And so welcome, Melanie, and the floor is yours.
Thank you very much. Wow, that was a nice summary. So yes, this is the result of my master thesis, and I finalized it during my PhD. But I'll first give you a little bit of an overview of, let me see, I didn't double click, no, okay, of what I will present to you. So of course, I'm just going to introduce the locality and the extinction a little bit, the methods that I used, of course, the results, and then a little bit of a discussion about why this matters and conclusions. And there's a lot of artwork in here, and most of it's actually from Mark Whitten, who's an amazing paleo artist. And without artists, we would have nothing to show because there are no pictures of dinosaurs, so I'd like to put them in. So I think we've all heard of the Cretaceous Paleogene impact. 66 million years ago, a bolide, or a massive piece of space rock, possibly from the Oort cloud, hit a reef just off the coast of the Yucatan Peninsula, and it caused the latest large mass extinction. It had a roughly 76% extinction rate, and it was very notably selective. So if you look at the period before the Mesozoic, then you've got all of these dinosaurs. They were the dominant group. And then if you look at the period after, they're gone. So, and mammals are still there, turtles are still there, crocodiles are still there. So it was quite selective in that sense, like on the macro fauna scale. So the impact occurred. It caused a massive shock wave to go through the crust of the Earth. It caused tsunamis, earthquakes, but also seiches, which I'll explain in a minute. But it also caused the actual meteorite itself to vaporize on impact because it hit a sulfur-rich coral reef. And what happened there is that the meteorite vaporized and the surrounding Earth rock melted and got fired into space. Some of it even circled the moon, and then it rained back down on Earth. And the Tanis event deposit in North Dakota was published by De Palma et al. In 2019. It contains what we call a seiche deposit. And a seiche is, in a way, it's similar to a tsunami, as in it's a body of water that shakes. But a tsunami radiates outwards from a single point in the ocean or against the ocean. And a seiche you can best compare to a pool during an earthquake. The shock wave actually goes through the underlying rock, and then the water that's on top starts slushing back and forth. So in this case, we had a remnant of an ancient sea in the middle of the United States. And because of this shock wave, the water from that sea went upstream into the Tanis River and over the Point Bar and back, and then again upstream and then back. And this mixed everything together: plants, marine life, terrestrial animals. And while this was happening, the impact spherules, so the pieces of Earth rock that melted, went out into space, and then later on came back down, they were coming down as this wave was hitting the area. And we find them, for instance, oh wait, I can turn on a pointer. Yes. For instance, here you see one, you see the sediments deform. I think it's actually even more visible here. You see the sediments deform, and then at the bottom, there's an impact spherule. And so this deposit is full of fishes, and I call them the direct casualties. There's tons and tons of other material in there, but I mean, if I find a dinosaur tooth, it could have shedded a month before the impact happened. And these fishes, they appear to have died on scene. And these fishes are actually buried according to their according to the direction of the flow. So for instance, you can tell that the wave was coming in and going back and coming in and going back. And the same goes for the fishes. They're coming in and they're going back. This one is slightly elevated as opposed to this one. And it's full of them. This was a massive luxury problem to be excavating. I've done excavations where you spend 2-3 days finding not a new single bone, and you're just sitting in 45 degrees Celsius, or what is it, 100 Fahrenheit or something, sweating and not finding a thing. And this was literally the opposite. We found a fish, but we could hardly get it out without damaging one of the other fishes that it was on top of or adjacent to. There were just so many fishes. And so I heard about these fishes because my former supervisor, Jan Smit, who's actually a quite well-known KPG boundary geologist, he presented these fishes, and I immediately said, the main difference here is that we're used to finding, first of all, marine records of the KPG boundary. Most of the known boundary sections are from marine records. And all of it is often low resolutions. It's at best a millennial or most often 100,000 year timescale. This is the final years in bone. So if we study these bones, can we find the season in which the meteorite impacted? So for this, I needed specific bones because, for instance, if a meteorite impact here in Sweden tomorrow and causes me to die, and you find a fragment of, I don't know, my humerus, my upper arm, and you study it, then you will not be able to find the season in which I died. I'm warm-blooded, but also my bones, they overwrite themselves when I grow. I stopped growing quite a while ago, so nothing new is being documented. So those are not practical things. But in the case of these fishes, they don't stop growing. They keep growing throughout their lives. And they are cold-blooded, which means they don't grow when they don't eat. They don't grow in winter. So, and the bones I picked, the dentary of the paddlefish, depicted here in pink, and the pectoral fin spine of the sturgeon, depicted here in pink, they grow what we say is, they grow appositionally. So like a tree, every year they deposit a new layer of bone on the outside. So I selected these bones. I could grab 3 dentaries and 3 pectoral fin spines. And then when you bring them home to the lab, they look like this, which is not very impressive, but they're small sections of bone. And I just want, before doing any analyses that could tell me seasonality, I wanted to make sure, are we still looking at the bone, or are we really looking at rocks? And for this, I'll describe a little bit what comes into play when something fossilizes, because when something fossilizes, what you often have is that there's a massive elemental exchange between the host rock and the bone. So for instance, you get leaching in of potassium from clay minerals into the bone, or manganese into the bone, or the worst is even if you get pyrite, which just will totally consume the bone, and then there's nothing left. So before doing any geochemical analysis, I wanted to make sure that there was no severe diagenetic alteration, which is what we call the process of the elemental exchange. And then I wanted to see, can we sample even these growth lines? Because you can see them here. This is a nice example of the paddlefish dentaries. You really see, okay, this is a sequence of growth, but there's also some staining, some discoloration, some blood vessels going through there. So it's not always easy to see. So we also wanted to use synchrotron tomography. So synchrotron tomography is when you use a particle accelerator, and with magnets, they send the particles round, so they don't collide, they just accelerate. But with every bending magnet, then the whole energy still fires in a straight line, the photons, and you can use them as very, very powerful X-rays. Not for you as a person, you will not survive, but for fossils, it's quite nice. So I used that to look at the really cellular level of these bones. And those are sort of the things we did. And even though our colleagues of the Palma et al. Had said, hey, these fishes died with impact spherules in their gills, so we know that they died between 15 and 30 minutes, we wanted to confirm this. So we also synchrotron scanned one of the fishes to see if it indeed had these impact spherules in its gills. Just gonna have a sip. So I'm assuming you've never looked at bone growth, so I'm just going to explain a little bit about bone growth. So as this bone grows appositionally, and this is a cold-blooded fish, I just gave you some directions here. So this is the inside of the bone, and the periosteum, that's the outside of the bone. So if a bone grows, new material is being deposited on this side. So the first distraction you always get is a blood vessel. So this is a blood vessel, and because it grows through the layers of bone, it really obscures something. So it's nice to have an idea of where it goes. You can always follow it in the bone, so that's nice. And as the bone grows, what it does is it makes osteocytes. It makes bone cells. And these osteocyte lacunae, so the spaces where the bone cells were, because it's dead, they're no longer there. That's what we call osteocyte lacunae. And in spring, or when the animal starts to grow slowly, that's when you have small osteocytes, not too many. And then when summer comes, you get more of them and they become bigger. And then when autumn comes, there's not that much food available anymore. Growth slows down, and there are less and smaller osteocytes. And then at the end of autumn, growth really stagnates. And that's when you get a sort of what we call a sort of lamellar growth. You get these layers, the annulus, we call it. That's really like at the end of the growth season. And then finally, when spring comes, that's when the line of arrested growth is deposited and the new growth commences. Excuse me. So the micro X-ray fluorescence, I cut thin sections, and actually the remaining sections of bone that you see below here were scanned to assess the diagenetic alterations. So we put them all side by side, and we observed where minerals are present, which minerals are present. We did line scans, we did total surface scans, just to see what the condition of these bones were. We also did stable isotope analysis, and that was the preparation actually took the longest amount of time, because I had to, here's a scale bar of 500 micron. So this is one of the sections of bone that I had to micromill out, and the computer was sort of almost exactly following my orders, but I had to keep correcting it, because every 2-3 slides, then the deviation became too big and I had to restart it. So I spent weeks and weeks doing this. And then I had to weigh 50 micron samples in a microbalance, and I spent I think 2 months doing that with a wet needle tip. I had to collect them. It was almost impossible to do. It was those were long days. Then the exciting stuff. I went to Grenoble, where they have the European Singertron, which is amazing. And I put my my sample on the sample stage, and then the X-rays came through here, and then there's a camera on the other side. And the specimen did what we call half acquisition. So it rotates 180 degrees, because that's enough. The camera will go through all the material then. And I was sort of lucky, because, so this is the fish that we scanned. Here you see the pectoral fin. And this is, so it's a paddlefish, but the paddle is missing. So here would have been the paddle. And then here would have been its dorsal fin and its tail. So it would have been up to here. So we had roughly 1/3 of the fish, but luckily, like the important bits, the head, the gill region, a little bit of the belly. And we could put it upright. And if you have like a big fossil and it's on a flat slab and then you rotate it, then the beam will still have to go through this enormous block of rock. And in my case, because we could put it upright and it wasn't that big, it was very nice and very easy to scan. It's one of the prettiest scans I have ever done. So the results of my thin sections. So just to give you an idea, you can already see some of the growth increments on just if you look at the overview, but it's always nice to zoom in where some of them can become really clear. In some bones, you've got what we call Sharpie's fibers. So you see that they have muscle attachment, but it actually makes it a little bit difficult to see the bone cells because of that, because it's another black line I have to look through. But these are my 6 specimens. And if I put 5 of them next to each other, then it looks like they all died just after forming a lag. But on this scale, it's quite hard to see the osteocytes. So we use the synchrotron for that. But first, the micro X-ray fluorescence. So just a total overview. This is a surface scan and all of the elements that are present there. But when it comes to diagenetic alteration, what you really look out for are stuff like yttrium, zircon, and they're not more. They don't exceed the background noise level. There was some measured iron. There was some measured manganese, so that's something to be concerned of. But then when we look at where it is present, then we find that iron here, Fe in green, in these images, and for instance, manganese here in blue is actually, it's in the blood vessels, which actually tells us that the preservation is really good because that's actually from the blood and it's sealed off the blood vessels and nothing has leached into the bone. There's some manganese in the sediment. So there's one section, that's this one with sediment. And again, the iron as well, it's in the sediment and it's in the blood vessels. Potassium is not in the bone, so no clay minerals were leached out into the bone. So this is really promising when it comes to doing isotopic analysis of confirming that I will likely measure the isotopes. And on this end, you see calcium and potassium and phosphate. Sorry, potassium is here. Calcium and phosphate. And calcium and phosphate are pretty much the main elements of bone. Bone is hydroxyl-apatite, and there's a potassium component and a calcium component. And in life, this balances out. And what we often see in diagenesis is that, for instance, manganese can very precisely take the place of potassium or of phosphate. I'm doing it again. And potassium can very well take the place of calcium. So then we would expect that these would not look homogeneous. And because they do look homogeneous, it tells us that there's no leaching out from the outside to the inside or whatsoever. And then the synchrotron scan. Let me see, can I turn them on? It should work. I'm just gonna go out of full, here we are. Yep, so at first we scanned the whole fish at a low resolution. I put the googly eyes in to give you an indication of where the front of his head is. And then we sort of digitally go towards the region with the gill cover. And then we did a higher resolution scan there. So this is the gill cover. So this is literally just the piece that's on top of it. And those are the gill arches. There's 5 of them because it's a jaw fish. Otherwise they have 7. And in the middle, we find these impact spherules. And these impact spherules are hollow. And that's because they crystallized or solidified in space. They didn't solidify on Earth because in that case, the lightest elements would always rise to the top, which they hadn't done. So these are the impact spherules, and actually one of them even, or at least one of them, still contained its unaltered core. So we could actually, if we were able to take it out of the fish, do some chemical analysis on this, but maybe in the future. So here you have just a view of what the scan looks like. What the scan looks like of the impact spherules. So I've circled them here in right, because they actually don't absorb any differently than the rest of the rock, but they are perfectly spherical. So you see them mostly visually. So when it comes to the segmentation, so the digital extraction of the bones, this was quite a job, but they were very satisfying to find. And also what we have are the ossified side walls of the brain of the fish. Further proving that we have very unique preservation here. So for instance, here you see the side wall of the brain. Here as well. They're actually, evolutionary speaking, also the oldest part of our skull, the side walls of the brain, which is also why fishes have them, because we're all fish, evolutionary speaking. So I've combined the osteohistology, so that's looking at the bones in all ways. So here we have, for instance, the thin section that I've showed earlier, and here we have the synchrotron scan, and I've just collected a couple of slabs on top of each other digitally. And in this case, I wanted to see the highest absorption. So I've maximized it, and here you see the minimum. And in the highest absorption, it's really easy to see the lines of arrested growth, so the winters. And in this one, it's really easy to see the bone cells, the osteocytes. And these green arrows, they point to where the really big ones are, so the good summers, I would say. And here as well. So here are 3 examples of those. And what we see is that they all appear to have died on the onset of a growth zone. Osteocyte volume was nowhere near the maxima that we see in earlier years. But okay, that's nice and cute, but that's qualitatively. So we wanted to see if we could quantify it. So what we did here was we actually segmented out these bone cells that you see here in 3D. And we've actually quantified how many there are and how big they are, so their volume. And what we see there is that we can really tell when they're growing and when they're not over several years. This is, in my opinion, the coolest part of our study, to be honest. And what we see here again is that, yes, it was increasing, but it was nowhere near the previous summer maxima. And then we did the stable isotope analysis. After, I think, 7 experiments failed, 1 finally did it. And I'll be very honest, I was expecting maybe more from oxygen. I was expecting that the variation between oxygen 16, the lighter oxygen, and oxygen 18, the heavier oxygen, we use it as an indicator for precipitation, for evaporation. So I would have expected that to give an idea of seasonality as well. But we're dealing with freshwater river that is not in contact, or it doesn't, there was no glacier filling it. It was a freshwater river, so there's not a high variance. What it does tell us is that our paddlefish was not a migratory fish. It didn't migrate between freshwater and marine water, because then these values would be all over the place. There were maybe some hotter and some colder periods, but still on this ratio, you can call this almost a straight line. Carbon, on the other hand, does do a lot more. And at first glance, I didn't really see much of it. But when you grab the same scale bar and you put it next to the other analyses, then there's suddenly a match, because every time we have a winter when the carbon values are lowest, we have a line of arrested growth. So every time it doesn't eat, so it doesn't get more carbon-13, it indeed doesn't grow. I'll explain to you a little bit. So carbon-12 and carbon-13 are the ones that we measure in this ratio. And carbon-12 is the lighter carbon, and carbon-12 is preferred by things that do photosynthesis to make sugar. So for instance, cyanobacteria or plants, they prefer carbon-12 because it's lighter, it's easier to make glucose out of. And that's how the food chain starts. But the carbon-13 is always present in the CO2 that they use. So what do they do with that? They put it into their exoskeleton. So you would have to imagine that that's a tiny ball of carbon-13, and the carbon-12 that's used as sugar, it just recycles in that tiny body inside. So if you were to eat that tiny ball, you're going to get more carbon-13. So that's how you can tell the food chain, because the higher up you get, the higher the carbon-13 gets. So this is somebody who eats somebody else. Awfully spoken, but that's how you can see, okay, it was eating, not eating, eating. And here we have some missing data because the analysis failed. That's sad, but eating, not eating, eating, not eating, eating, not eating, eating. And this is death. Nice dotted line. And also I've just visualized again the growth marks, so where they were growing, where they stopped growing, and then the lag at the end. So on a spring day 66 million years ago, a bolide impacted into the present-day Yucatan Peninsula and ended the era of the dinosaurs. But why does that matter? Well, if it was spring in the northern hemisphere, then it was autumn in the southern hemisphere. Spring, I think we're all sort of enjoying it right now, is the onset of growth. The birds are going at it, and you get flowers, new leaves, everybody's out and about looking for food, everybody's vulnerable, I would say. And in autumn, the opposite is the case. Plants are already shedding their leaves, mammals are burrowing, preparing for hibernation, fattening up for winter, and making sure that they'll be comfortable for the cold period. And then this catastrophe hits, and it hits the two hemispheres in a completely different stage of their biological life cycles. And if you then look at the post-recovery, the post-impact recovery phase, then it appears that in the southern hemisphere, this was over twice as fast. And that could just be because in the southern hemisphere, where even these birds actually survived the KPG extinction, in the southern hemisphere, these birds they hadn't just laid their eggs that needed to incubate for I don't know how long. No, they were sheltering, or they were hibernating, and the same goes for mammals. There are some actually some crocodilians that survived on the southern hemisphere, some turtles that survived on the southern hemisphere. The only problem is that we don't have that many fossil records from the southern hemisphere of the latest Cretaceous, so it's hard to tell exactly which animals survived, but it could just be that in the southern hemisphere, because they were preparing for winter, the tragedy was not as unfortunately timed as in the northern hemisphere. I have a lot of people to thank for this because I did not do this alone, of course. So I have my co-authors, Jan Smit, Dennis Voete, Camille, Paul, Sophie, Koen, Suzanne, and Jeroen, and my current supervisor, and everybody else who helped me along the way. And thank you very much for listening. And if you have any questions, ask them all.
Great, thank you. That was really great. One of the bad things about Zoom is we can't really show our appreciation, so it always seems a little lukewarm. But that was really, really fantastic.
Thank you.
Yeah, I guess we're open for questions.
Yeah, we have a brief time for questions. So speak up if you have questions. So I know it's not your work, but there were also mammals found at the site in burrows. Could you tell us about that, which goes along with your interpretation, I believe?
I heard that there was indeed burrows found that were underneath Tanis, but I'm actually not sure if they predate the deposit or not, because that's the thing about burrows. Because if you make a burrow today, the host rock you're burrowing in can be Cambrian in age. I mean, so it's very hard to date. I believe I recommended them that they scan the burrow and see what was inside, but I've not yet heard any outcomes of that. But it could very well be that it's a burrow that is Paleogene which I think is most likely actually. But the main thing is that if you're on the surface when this thing hit, you were likely either directly evaporated or burnt, or I mean, you were extremely vulnerable. So if you were underground or underwater, you had already, if you survived that first punch, then you're already at least a tiny bit better off. So there were many animals in the northern hemisphere as well as in the southern hemisphere that were capable of burrowing or hibernation or any state of torpor where they like lower their bodily functions to survive harsh periods. But it helps if you were already doing that. And most animals in spring were not.
So for the audience, I should say, feel free to raise your hand or just unmute yourself and you can come in with questions. I did have one too while we were waiting for people to collect their thoughts. So this is an ignorant question. So this is very elegant, the way you sort of, from the bone growth, you could tell the season. Is this something that people have done before, or is this like a new technique that you had to invent for this study?
I would say that the combination of the techniques used is new, but none of them were invented by me. So I did isotopic analysis on different bones, different animals, and I know you can actually, in some teeth actually record daily resolution of growth. So a higher resolution could even be possible, but these fish didn't have those teeth, so didn't have teeth. So that was not an option here. But I would say all of the techniques I used are definitely not come up by me, but I did grab every chance to make sure that I could have an idea of the preservation and then reconstruct the latest years of the Cretaceous and hopefully find the time of the year that it struck.
Yeah. Well, I guess in a way that's better that this is a well-established technology, because then, this really important conclusion doesn't have to turn on an untested technique.
Yeah.
So
I think the only thing that was actually new, I wouldn't say, but Suzanne and Jeroen, so my final two authors, they had just developed this method for the mass spectrometry, so for the isotopic analysis, to temporarily freeze the CO2 that we were measuring. Because when you do isotopic analysis, the sample heats up, and that's actually also why you lose a tremendous amount of your sample when you're doing the process. And my samples were too small for that. If that would happen, I would get no amplitude anymore. So they temporarily froze it. And this was a technique that they were actually using or developing at that time. So one of my analyses failed because it didn't yet work, and then one of my analyses worked. But in the process of writing up the paper, they already wrote up a paper describing the method. So that helped that I didn't have to do that. Yeah.
Well, actually, I should let other people come in, but I just want to ask one more follow-up thing. And then I see Patricia has a hand too, which is, are you looking for something with teeth then? Is that something that,
Oh, I'm definitely always working on teeth and interested in teeth.
But I mean, at this site at tennis?
I don't know if I will get to go back. I would love to go back and do more with tennis, but for now I'm working on some of the scans that we have of this fish, of the paddlefish, and describing it because it's a new species, new genus, new fish, and it deserves description. And after that, I don't know actually.
Okay, very good.
Time will tell.
Yeah.
I just wanted to say, I mean, that's terrific to hear about this new technique. And yeah, you're putting together all of these things, some of which are pretty recent. Paul Taffero has been increasing the number of paleontological specimens that it's sort of like his pet project there at Grenoble at the synchrotron. And so it's not brand new, but it was a pretty limited number that could be scanned until very recently. So you really are right there on the cutting edge. Synchrotron isn't new, but applying it to fossils is fairly new. And then applying it to more fossils is something that's really only just become pretty available. People here, some of them know Isaiah Nengo and the skull that he found, Lessie. This was one of the earlier ones that was done on that. All of those scans you saw, it's the same thing. that she's using, and the same people that are promoting it. So it's nice to see physics and paleo, working together.
Yeah, it did a lot for my physics knowledge, actually, because that was sort of slowly deteriorating over time. And then I got to use the synchrotron. And then, yeah, I mean, when you're there, you want to know everything about how it works. So it's amazing.
Yeah.
David Gurari, I think you had a question, maybe?
Thank you for delightful talk. I'm curious, do we know much about climate 66 million years ago? Was it highly seasonal, north-south?
That's a very good question. So I actually didn't mention that in the presentation, but before I started this project, I first needed to make sure that there were 4 seasons, of course, because otherwise the project wasn't really useful. But so in the Hell Creek, where we did the excavation, so the Northern Hemisphere, they had winter lows of roughly 4 degrees Celsius, 4 to 5 degrees Celsius, and summer highs of 19, 20 degrees Celsius. With a seasonally wet period. So they had 4 seasons and a relatively long period of growth. I think the biggest difference was because the Atlantic was, of course, already open. Some of the continents that we now know are higher up were still below the southern half of Africa, like Madagascar, India, Australia. We're very close to Antarctica still. But there were no ice caps on the North and the South Pole. I think that was the biggest difference. So you didn't have that high temperature gradient from North Pole to South from poles to equator. But there were 4 seasons where I excavated this material.
Thank you.
So I think there's a question from Patrick Mason.
Hello. There we go. So I was writing down my question, so it's kind of half formed at the moment. After you publish this work, what would you like to do next or go from here?
Oh, good question. I'm actually very eager to use these techniques. So not necessarily on the KPG extinction, but there are quite some extinctions, but also like mass graves of fossils, and it's often that we don't know if they died at the same time, if there was some sort of catastrophe, or if this was a gradual accumulation, like for instance the La Brea tar pits. We know for a very long time animals got stuck in and died, but there's like the Hangenberg event and Devonian extinction. That I would like to see if I can reconstruct roughly time of death and if the different individuals died simultaneously or not. But I'd also like to expand on this and maybe try and find causes of death for other animals. So I'm definitely certain I want to use these techniques for more things, but I'm not necessarily hung up on having to do the same thing.
You should come to my site in Africa, that in Turkana, where we have sediment, 100 meters of sediment that was laid down in perhaps sometime within 200,000 years all at once.
But then if it's Turkana, then this name should ring a bell. Because I think Jeroen actually does a lot in Lake Turkana as well. That's my former supervisor.
Bring him along.
Oh, I will. I'd love to.
Grant has a question.
I was wondering, as far as like what would be like some potential future directions to kind of continue this study? And then I was also wondering, is there a potential for correlative evidence regarding plants, or would it be necessary to have those cold-blooded fish in order for something like this to be evidenced?
I mean, for me, I'm a vertebrate paleontologist. So for me, I work best when I look at bones. But that doesn't mean that plants are less informative. Not at all, actually. I didn't mention that here, but I also found a sprouting seed. The only problem is I cannot prove it's a fossil. And if I cannot even convince my co-authors, then I cannot convince anybody else. I would have to do a very extensive C14 dating to find out if it's too old, and in that case, but it does fit my picture, of course, that it's spring because it was sprouting. I actually think that there's a lot more in plants, because when it comes to extinction, don't forget that most extinctions are actually, it goes from the plants up. So first you lose the primary producer, and then you've got the whole food chain collapse. So yes, don't underestimate the plants. I would say they're highly useful. And when it comes to studying and trying to understand this extinction better, that's definitely a sort of untapped resource. But also the Southern Hemisphere is almost blank territory when it comes to data. There is way too little data. And if I can personally add there, the last thing the Southern Hemisphere needs is for us in the Western world to put on our hats and go dig there and take the stuff away. I would like to see a bit of a more incorporative approach there, because they've got their expertise and they've got people that can excavate and study them, but they often don't have the funds to publish open access in Nature, or to describe or analyze the material. So I would say that when it comes to like the flora and fauna of the latest Cretaceous of the Southern Hemisphere, there's massive terrain to be gained.
Thank you.
I guess I had one more question, and I don't know, I think we're also getting to a point where it's getting quite late for you.
It's time, but one more is
but
fine.
Since there were so many questions about the future, can I ask a question about the past? Like, could you tell us a little bit about how you got here? Well, not the deep past, but about your past. I think we might have undergraduates or even younger students maybe in the audience, and they might be curious about how you got into this field. And
so I actually started because I was collecting shells on the path from my dad's house to my middle school as a child, and I was collecting them, and then one day I found one that was heavier than the others, and it was like it didn't shine as the ones you find on the beach, and it was really hard. So I took it to the library, and the librarian helped me to the geology and biology section, and I found out it was a fossil.
Mm-hmm.
This was and it was extinct, and it could be dated to this period millions of years ago. And I was like 9 or 10. And I was like, wow, that's amazing. And you have to understand, I'm born in 1989. So for some of you, I'm young. For some of you, I'm old. But this is I was exactly the age group that was too young for Jurassic Park. I wasn't allowed to see them. I didn't even see the advertisements. It wasn't aimed at me. I was too young. So I missed the hype. I was well behind the hype. Yes, I had seen Land Before Time, which is amazing, but it was a cartoon. But it wasn't until I found the fossil as a child that I realized this is really cool. And so I studied earth sciences for multiple reasons. It wasn't just fossils that interested me. I also thought minerals were very amazing, and volcanoes. But then I realized if I would do volcanology, I would probably get too close to the edge one day and not be able to tell afterwards. So decided maybe, because I'm a bit of a daredevil, so maybe not test fate too much. So I decided to back out of that. And also I was trying to do the calculations of when magma solidifies and which minerals crystallize. And I was getting stuck on the math. It was not my strongest not my strongest skill there. So but I mean, to be a paleontologist, you can do 2 routes in pretty much. You can take the geology or earth sciences route, and you can take the biology route. Mm-hmm. But either route you take, you need the other bits too. So I took the earth sciences route, and while I was doing that in my free time, I was drawing skeletons and trying to learn all the names of the bones, because we're all vertebrates, so we all have the same system. I jokingly always call it the 1 to many system. Every limb has 1 bone, 2 bones, and then many bones. And that's all the vertebrates have them. Some have lost some of them, but that's the bow plan, the body plan that we have. And once you get to know that, then yes, you can go into the field, excavate something, and be like, that's a femur of a triceratops, which I had never seen before. But I could recognize that it was a femur, because you know a little bit about the bones. And that's something that really pulled me in. And yeah, the more I find, the more questions I have, and the more questions I try to answer, the more questions I get. And I would say my biggest advice, if you want to do something like that, you have to be a little bit crazy. You have to be a little bit crazy about it, because it helps. It motivates you when times are tough. Yeah.
I'm so glad that you said that about drawing, because some of my introductory evolution class students are on, and I have them draw things and they're like, what? So there you have it in the flesh, not just old codgers, not just Louis Agassiz, but young people doing their theses on cool subjects are drawing things. So well done. Thank you so much.
Thank you for having me.
Yeah,
Yes.
well, that's really been a lot of fun. I think there's lots more to talk about, but
Yeah, I hope you'll come back.
Wrap up here. Yeah.
I might. I've also recently signed a book deal, so which is still a bit surreal, but I started. I'm now on chapter 2. So
Excellent. We look forward to reading it.
Awesome.
Okay, well, we'll have to have her come in person when the book comes out so we can have a book signing.
Ooh, oh wow, that would be nice. I'd love to.
Okay, well, everybody, I encourage you to attend on Thursday evening as well. We'll have the video, and then we'll also have more discussion. And we will attempt to answer questions, even if Melanie's not there.
If you leave Thursday evening with like really important unanswered questions, please just send me an email and I'll try to answer them.
Perfect. Yeah, excellent. Excellent. Okay, and I hope to see everybody tonight at 6 p.m. for the pandemic. Take care, everybody. Thanks so much. I'm really glad to see the good turnout. So,
bye.
Thanks for having me. Bye-bye.
Yeah, bye. Great to meet you.